All right, welcome. I'm Mark McClellan. I'm the director of the Duke Margolis Center for Health Policy. On behalf of the center, I'd like to welcome you all to this public meeting on minimal residual disease as a surrogate endpoint in hematologic cancer trials. This meeting is being convened under a cooperative agreement with the Food and Drug Administration and in collaboration with the Office of Hematology and Oncology Products. And we're very pleased that all of you could join us today, and also for those who will be watching online. Today's workshop is going to focus on advancing the discussion around the validation and use of minimal residual disease, or MRD, as a surrogate endpoint in clinical trials for hematologic cancers. As many of you know, the standard endpoints for clinical trials of new therapies for hematologic malignancies have included measures such as overall survival or progression-free survival, event-free survival, and response rate. Over the last 2 decades, however, the good news is that advances in cancer research and drug development have transformed the cancer landscape, including for many patients with these hematologic malignancies, and the introduction of the novel therapies to treat these diseases has significantly improved clinical outcomes. The result is higher rates of remission, longer remissions, and better survival rates. Some of these cancers that were once rapidly fatal can now be managed more like a chronic disease. But as these outcomes improve, the amount of time required to measure endpoints like overall survival or progression-free survival has also increased, leading to longer and more expensive clinical trials. For this reason, there's been a lot of interest in identifying and validating new surrogate endpoints that can be measured at an earlier stage and which could be used to support faster clinical development and regulatory review of further innovations in therapies for these cancers. At the same time, there have also been advances in molecular testing to enable the detection of leukemic cells with much greater accuracy, improving assessments of how deeply patients respond to a given therapy or combination therapy. Accurate measurement of these remaining cells, which is minimal residual disease, can potentially be used to guide decision-making across a range of contexts. In certain clinical care settings, for example, MRD status can enable better treatment decisions, allowing physicians to assess a patient's prognosis more accurately, identify patients who are at risk of relapse, or determine if they've responded well enough that they no longer require treatment. But there are still ongoing questions over how and to what extent MRD assessment should be integrated into routine clinical care, as the strength of the evidence base to support its use in guiding treatment decisions varies across hematologic cancer types and subpopulations of patients. For drugs intended to treat hematologic cancers, MRD is also a candidate surrogate endpoint that could be used to develop evidence for efficacy and regulatory review. Validating MRD as a surrogate endpoint could have real consequences for accelerating the development of new treatments for patients suffering for these cancers by shortening that development and review time. But the process of validating a surrogate endpoint is more complicated than just demonstrating a correlation between a surrogate such as MRD and a clinical outcome that matters, like overall survival. As we'll talk about today, you'll hear about a lot of discussion on what needs to be shown for a surrogate to reliably and precisely predict the treatment effect on the corresponding clinical endpoint to a degree that satisfies the scientific criteria and the regulatory review criteria, and how this may vary across different clinical sites and contexts, how this needs to be demonstrated across multiple clinical trials. Furthermore, even when a surrogate has been validated in a particular context, it's not necessarily appropriate to extrapolate that determination of validity to another disease or patient population or therapeutic context. The existing evidence to support the validity of MRD as a surrogate endpoint varies considerably across the hematologic cancer subtypes, as we'll hear about today, owing in part to an underlying molecular and pathophysiological differences between these diseases. A lack of standardization in how MRD status is measured, both between individual studies and across different institutions, creates additional challenges by limiting the ability to compare study outcomes or draw meaningful conclusions between different studies. So collaborative efforts to standardize these various methods are underway, and we're also going to hear about some of those today and how they can help address the challenges that we're discussing. Between 2012 and 2014, FDA convened a series of workshops to address issues like these in MRD use in 4 cancers: multiple myeloma, acute lymphocytic leukemia, chronic lymphocytic leukemia, and acute myeloid leukemia. Since that time, there has been significant progress in developing evidence to support the use of MRD as a diagnostic and clinical tool in each of these cancers. However, questions remain over how best to move forward in terms of its validation as a surrogate endpoint that can be used to support regulatory review. So that's the main reason we're here today. The Duke-Margolis Center for Health Policy is convening this public workshop to address the current state of the evidence available to support validation of MRD as a surrogate endpoint and advance the discussion around its use in developing new treatments of multiple myeloma, chronic lymphocytic leukemia, acute lymphocytic leukemia, and acute myelogenous leukemia. Specifically, in the workshop today, we hope to discuss the regulatory background for use of MRD as a surrogate endpoint for regulatory decisions, and discuss the statistical basis for demonstrating and validating surrogacy, and then present the evidence available to support MRD for clinical trials in new treatments of multiple myeloma, chronic lymphocytic leukemia, ALL, and AML. Before we get started on the actual content of our event today, I'm going to turn to Greg Daniel, the Deputy Director at our center, to go over the agenda and also make a few logistical announcements. Greg?
Great, thanks, Mark. Allow me to echo Mark's thanks to all of you for joining us today, which should be a very interesting but also an important discussion. We'll begin the workshop with a presentation by Vishal Bhatnagar and Jennifer Dickey from the FDA, who will provide some context and define the goals of the public event today and provide some of the regulatory background on in vitro diagnostics issues related to hematologic cancers. The subsequent presentation from Dan Sargent of the Mayo Clinic will also go into more detail on the issues that are the focus of today's meeting. He'll provide an overview of the biostatistical considerations for demonstrating surrogacy. And then we'll take a quick break at 10:45. After that, and for the rest of the day, we'll break out into 4 one-hour sessions. And each of the sessions will explore the state of the evidence surrounding the use of MRD, in particular blood cancers, as well as the remaining barriers that will need to be addressed in order to move the discussion forward and make progress. Session 1, for example, will focus on multiple myeloma. Session 2 will focus on chronic lymphocytic leukemia. And then we'll take a quick break for lunch at 12:45. After lunch, Session 3 will focus on acute lymphocytic leukemia, and then Session 4 will focus on acute myeloid leukemia. I would like to also note that Session 4 will be moderated by David Rosari from the Duke Cancer Institute, who we're pleased to have join us today. You'll also note in the agenda that each session will begin with an opening presentation, a set of panelists who will provide brief overview and comments from the presentation. And then each session we have designed some time for moderated discussion and participation from all of you in the audience. So as you hear the presentations and the panelists, please make sure that if you have questions, comments, or suggestions, to make sure you get those addressed. We have microphones at each of the tables, so when we go to the Q&A session, we'll have Mark, myself, or David Rosari call on you. Also, as a heads up to the panelists, we do have staff in the front who has a time card. We have a lot of panelists for each session, so she will be very judicious in making sure that we end on time in each of those sessions. Lastly, I would like to inform you that this is a public event. We are live broadcasting via which will be on the web. So the comments that you make will be part of the public record, but we hope you feel free to bring your issues, comments, concerns, suggestions to the discussion today, because this is a big important part of moving this discussion forward. And we want to make sure that we have captured all of your thoughts and ideas as we move forward. And now lastly, lunch will be on your own. We do have coffee and drinks in the back, so feel free to help yourselves to those throughout the day. Lunch will be on your own. We have a number of restaurants in the local area, and I think that for all of you who registered online to attend today, I think we did send an email that had other suggestions for restaurants nearby. You're also welcome to bring any of the food back to the room here today for the remainder of the day. If you have any questions about today's discussion or the logistics, please feel free to reach one of our staff members out in the hallway. And with that, I'd like to kick things off by introducing Vishal Bhatnagar, Medical Officer in the Division of Hematology Products at FDA. He'll provide the FDA perspective on using surrogate endpoints in regulatory decision-making and help establish the context and goals for the workshop. Thank you.
Good morning. It's my pleasure to participate in this meeting regarding minimal residual disease as a surrogate endpoint for clinical trials in hematologic cancers. My name is Vishal Bhatnagar, and I'm a clinical reviewer in the Division of Hematology Products in the Food and Drug Administration. The purpose of this presentation is to provide the regulatory background, specifically the approval pathways, to set the stage for MRD as a surrogate endpoint. For a new drug to be approved, the Food, Drug, and Cosmetic Act requires that the application must contain substantial evidence of efficacy with demonstration of acceptable safety and adequate and well-controlled trials. The FDA examines the strength of evidence in the context of the disease state, available therapy, study design, and endpoint selected. The application must contain enough information to allow for the generation of a product label that defines an appropriate patient population and provides adequate information for the safe and effective use of the drug. There are two pathways for approval. For regular approval, the approval is based on demonstration of clinical benefit or an effect on an established surrogate. Clinical benefit is defined as a measure of how a patient feels, functions, or survives. Accelerated approval is an approval mechanism designed to expedite drug development and allow for the review of new drugs for the treatment of serious or life-threatening conditions. For accelerated approval, a therapy must provide meaningful therapeutic benefit to patients over existing treatments. And approval in this case is based on an effect on a surrogate endpoint reasonably likely to predict clinical benefit or a clinical endpoint other than survival or irreversible morbidity. Importantly, there is a requirement for post-approval trials to verify the anticipated clinical benefit. The design of the confirmatory trials is proposed by the sponsor and agreed to by the FDA. The clinical trials must be carried out with due diligence, and they must be adequate and well-controlled. The agency has allowed post-approval clinical trials to be conducted in a related disease setting. Typically, sponsors conduct their post-approval clinical trials at an earlier disease setting. Let's take a moment to define what a surrogate endpoint is. A surrogate endpoint is a measurement that can be used to define a measure of direct clinical benefit, but ought to be validated statistically to correlate with a true clinical endpoint. The strength of this surrogacy is a variable between diseases and depends on the biological plausibility of the relationship. Furthermore, evidence from epidemiological studies and clinical trials is required to understand the prognostic value of said surrogate, and whether interventions cause a corresponding effect on the surrogate and the clinical benefit measure. Essentially, a correlate does not make a validated surrogate. Therefore, strength of surrogacy must be established. For example, a biomarker is a type of surrogate endpoint that predicts clinical benefit and can be objectively measured as an indicator of a normal process, pathologic process, or response to an intervention. Ideally, the biomarker captures the full impact of the intervention on the true clinical outcome seen at the top of the diagram. A historical example is HIV as the disease, with the intervention being highly active retroviral therapy, and the biomarker as HIV viral load as a surrogate for survival. In that case, HIV viral load is on the causal pathway of the disease and is relevant to the natural history and was subsequently fully validated as a surrogate endpoint. In suboptimal cases, the intervention may not affect the biomarker despite correlation with the disease and the ultimate clinical outcome. This is illustrated at the bottom of the slide. In this case, a biomarker may change with the disease burden as illustrated by the dotted line, but is not impacted directly by the intervention. In this case, the biomarker does not adequately capture the full impact of the intervention or disease process. For this reason, it is imperative to understand the underlying disease and how the biomarker fits in that pathway. These are unofficial definitions and examples designed to clarify the distinction between the two types of surrogate endpoints in the two approval paths. The discrimination is important for determining the appropriate approval pathway. The difference between an established surrogate and an unestablished surrogate is multifactorial, but is really based on the strength of evidence supporting the surrogate endpoint as a predictor of clinical benefit. Later this morning, Dr. Sargent will discuss major biostatistical considerations for determining surrogacy in greater detail. Generally, sponsors are required to meet with the specific review division to ensure that the appropriate regulatory pathway is being pursued based on the strength of evidence and pertaining to the surrogate being used. Revisiting the two types of approvals, both regular approval and accelerated approval pathways can be based on surrogate endpoints, but there is a difference in the type of surrogate appropriate for each pathway. Regular approval can be based on an established surrogate endpoint, while accelerated approval is based on a surrogate endpoint that is deemed reasonably likely to predict clinical benefit. Therefore, endpoints may be different in the two pathways even for the same disease. For example, a drug may be granted accelerated approval based on overall response rate, and in a confirmatory trial, receive regular approval based on PFS, despite the fact that both ORR and PFS are surrogate endpoints. This is a non-exhaustive list of recent accelerated approvals in hematologic malignancies. These are therapies that demonstrated meaningful therapeutic benefit to patients over existing treatments and were based on surrogate endpoints reasonably likely to predict clinical benefit within their disease settings. Practically speaking, these are therapies that were studied in single-arm trials with primary endpoints of overall response rate. Some of the drugs that were on the prior slide appear on this slide. For example, ibrutinib was granted accelerated approval in chronic lymphocytic leukemia based on overall response rate, but was subsequently given regular approval after demonstration of a PFS benefit in the same disease. This is an example of conversion from accelerated approval to regular approval after demonstration of an effect on an established surrogate. Generally speaking, for regular approval, clinical benefit or improvement in established surrogates are necessary. A common established surrogate for hematologic malignancies is progression-free survival. For accelerated approval, tumor size reduction measured by overall response rate is frequently used as an endpoint. In the pursuit of further expediting drug development and approval, the purpose of today's meeting is to discuss the validation and use of minimal residual disease as a surrogate endpoint in clinical trials for hematologic cancers. In this context, the next speaker is Jennifer Dickey from the Center of Devices and Radiological Health, who will discuss the topic of in vitro diagnostics and CDRH's regulatory perspective on MRD assays. Thank you.
Thanks very much, Vishar. If Jennifer, would you like to come on up? Just a brief introduction. Jennifer Dickey is a senior regulatory reviewer in the Office of In Vitro Diagnostics at the at the CDRH. She'll provide an overview of the issues facing in vitro diagnostics for hematologic cancer and help set the stage for today's discussion too. Thank you.
Oh, it's just the same slide deck? Hello, thank you so much for having me here. My name, as was said, is Jennifer Dickey. I work in the Office of In Vitro Diagnostics and Radiological Health in the Center for Devices. Our office, I'm in the Division of Molecular Genetics and Pathology, and our primary focus is on co-development kind of things in oncology, such as companion diagnostics and other sorts of indications. So to go back to just set the stage, I'm deriving a lot of my definitions and discussion points from the BEST biomarkers, endpoints, and other tools resource that was developed in collaboration with the FDA and NIH and other thought leaders when, in the course of discussion around these very intricate topics, it was realized that there is actually a lot of variability in the field about what is a biomarker, what is an endpoint, what is a medical device, when does a biomarker become a medical device. These sorts of questions actually lead to a lot of confusion in the field. And so just starting back at square one, a biomarker is a characteristic that is an indicator of normal biological processes, et cetera. But a biomarker is not necessarily a medical device. So an assay is the analytic procedure for detecting or measuring the presence, amount, state, or function of a biomarker. So if you have an assay, you're assessing a biomarker, and one component of that test of an assay is the test tool or instrument. So in the sense of where I fit into the picture, where medical devices lie, we look at the test tools or instruments. So it's an assessment system comprising three essential components: the materials for measurement, the assay for obtaining the measurement, and the method or criteria for interpreting those measurements. And so when an applicant comes to CDRH, we look at the intended use of your medical device and we ask, what are you measuring? How are you measuring it? And what will you interpret from that measurement? It's the entire system of not just, and that's why frequently in interactions, when we talk about minimal residual disease, we have to step back a little and say, what's your definition? Is it blood, bone marrow? What kind of assay technology or platform? What's your cutoff? How are you going to interpret it? What decision making will take place because of the result of this assay? Is this larger systemic review that the IVD looks at? MRD necessarily is not necessarily a medical device until you start getting into the assay, the tools, and the instruments, and the interpretation. And what we ask when we talk about medical devices is validation, establishing that the performance of your test tool or instrument is acceptable for its intended purpose. The elements of validation include, but are not limited to, analytical validation and clinical validation. I'll go into this in a little more detail. So surrogate endpoint biomarkers are distinct from medical devices. Surrogate endpoint biomarkers and other biomarkers are not necessarily medical devices, but it is to your advantage. And in frequent interactions with sponsors, we very helpfully try and guide you towards identifying a single consistent tool. This would include consistency around the materials for measurement, the assay for obtaining that measurement, and the method or a criteria for interpreting those measurements. Measurement at what time, around what decision-making threshold. This generates interpretable data in multi-site trials. It generates data that is able to be leveraged for larger decision-making. And surrogate endpoint tests generally become monitoring assays if successful. So they become medical devices in the course of their validation if it becomes that they are used in clinical use later for monitoring patients while under treatment. And so these generally become monitoring or prognostic tests. So the role for biomarkers in the agency is varied. Actually, every center in the agency has a biomarker office, but focusing on the Center for Drugs and the Center for Devices, the Center for Drugs works in the drug development tools, those tools you use during clinical trials, and in the biomarker qualification program. CDRH likewise has medical device development tools for clinical trials for approval or clearance of medical devices, and also in vitro diagnostics and companion diagnostics. And this is where I'm going to focus today. But I'd like to emphasize that we work together a great deal. CDRH consults with CDER and vice versa to understand the clinical and analytical validity needed for tests of biomarkers for clinical purposes. And so we interact back and forth quite a bit, and we are happy to interact more or field questions, and we're happy to interact with our CDER colleagues while we work towards goals in MRD. So shifting towards in vitro diagnostics. The standard review process for an IVD is determined by its risk classification. If it's moderate risk, it would be deemed class 2 and move through what's called a traditional 510K pathway, which gives FDA clearance, not approval, but the standard is that the device shows substantial equivalence to a legally marketed predicate. And so if you have an assay that does something that's already cleared, you just have to demonstrate substantial equivalence to that first assay to get clearance or approval. If the assay is high risk, it's designated class 3 and requires a pre-market approval application or PMA. The FDA approval process for this is that the device demonstrates safety and effectiveness, very similar to the drug standard for NDAs. If your assay happens to be moderate risk, but there is no predicate device, then you would move through this pathway here, the de novo 510K, where there's no predicate, but you demonstrate safety and effectiveness, and with the use of special controls that are published in the CFR, become regulation, we allow the clearance and marketing of this assay as class 2 moderate risk, and then your assay can be a predicate for future assays. So the basic components of an IVD submission include, most critically, the intended use. This would include the analyte, the instrument, the interpretation, the use. The device description, any specimen handling or pre-analytical requirements, analytical performance, clinical performance, instrumentation, software as applicable, labeling, and for PMA or high-risk products, we do review in the pre-market setting manufacturing, design controls, and quality systems. These things are required for all products, but pre-market review is only needed for high-risk products. So what does FDA review? We review the sample collection, the instrument or assay, the analyte extraction, and the software or interpretive aids that are used in the device. So for analytical validation, the question is, does the test measure the analyte accurately and reliably in the hands of the intended users with various sources of variability? Generally, for analytical performance, we look at the sensitivity, linearity, the limit of blank, detection, quantitation, the specificity of your assay in terms of cross-reactivity, interference, exclusivity, the accuracy as compared to a valid comparator or gold standard reference method, and the precision. Can you get the same result reliably if you repeat it? And in a multi-site reproducibility, could you get the same response? And we generally look at performance around the pre-specified clinical decision-making threshold. And this is why the clinical decision-making threshold should be pre-specified ahead of this analytical validation activity and is a key element to designing your medical device. For clinical validation, The question is your study design appropriate to support your intended use? Is it for diagnosis, disease subclassification, monitoring, determining the risk of disease or prognosis or prediction? And do the test results correlate with the result from the predicate device or from the expected clinical endpoint? So the example I have for today is the Assuragen BCR-ABL kit, which was approved as a de novo. The link below is the link to the decision summary that contains all the analytical and clinical validation information that was submitted for our review. And as you all know, in CML, kind of early in the process, it was understood that achievement of a major molecular response predicts improved progression-free survival. And so this became a very valuable monitoring or prognostic tool in CML. And so when the Assuragen BCR-ABL kit came in, it was their undertaking to demonstrate that their assay, their specific assay, could recreate this or recapitulate this clinical benefit using their analytes in their intended use. So notice their intended use is an in vitro nucleic acid amplification test for quantification of BCR-ABL and ABL in total RNA from whole blood in diagnosed CML patients expressing the transcripts, the major transcripts. So its results are expressed as a log molecular reduction value or on the international scale during monitoring of treatment with tyrosine kinase inhibitors. Notice that minimal residual disease is not present in this intended use. This is a prognostic monitoring assay. So the analytical studies that Assuragen provided were clinical sample pools at 5 transcripts levels, MR1, 2, 3, 3.5, and 4, knowing that the clinical decision-making threshold was MR3, in this case, major molecular response. They demonstrated linearity from MR 0.1 to 4.8 using clinical samples diluted into normal whole blood. They established an LOD and LOQ of MR 4.7, which is well below the clinical decision-making threshold, and established RNA input ranges between 1,000 and 5,000 nanograms. For their clinical studies, generally in IVDs, you don't need prospective clinical studies. You're trying to recreate a clinical state. And so it was a retrospective multi-center clinical outcome study at 3 U.S. sites to establish the clinical validity of MR3 for their assay. They're not trying to recreate MR3 as the standard. They're just trying to say that their assay at where they read MR3 is accurate and reflects the clinical literature. For MR3 for event status at 36 months in CML patients. So they had a total of 139 samples from 98 patients collected at 2 sites. And the inclusion criteria were that the samples must be in the 12 to 18-month time frame. And notice the time frame. So patients assessed at 12 to 18 months, and then you look at outcome in terms of their clinical status at 32 to 40 months. I'm sorry. The samples were from adults diagnosed with CML, and after the date of the sample and through 32 to 40 months, clinical treatment failure and progression, disease progression information needed to be acquired. And so the clinical study outcome evaluation was event-free survival at 32 to 40 months on samples tested 12 to 18 months after starting their therapy, and device performance was assessed by the probability of at least one event by endpoint 32 to 40 months after initiation of treatment. And the acceptance criteria for this were that the two MR3 yes or no would have estimates differing by at least 10 percentage points, and they succeeded in their endpoint. So the test was granted approval. It can now be used as a predicate assay for similar intended uses. And just to clarify, the special controls for this assay is it's for indications for use based on MR3. Device performance from either a method comparison study to the predicate or through a clinical study can be used to get clearance or approval for a similar assay. For indications for use not based on MR3, like say you wanted to do a study around MR4, MR5, something of this nature, you would need to provide evidence from a new clinical study. Device reproducibility data needs to be generated using a minimum of 3 sites, at least 2 of which must be external sites, 2 operators at each site, 3 runs per operator, non-consecutive days, 5 different BCR-ABL concentrations. They're well distributed over the measuring range. And they must demonstrate traceability to the WHO reference panel with a multi-point calibration. So these are basically how the agency specifies the requirements or the performance characteristics that should be provided to us in a 510(k) submission to claim substantial equivalence to this assay. And so it's these sorts of models, the time at which you take the measurement, the time at which you assess the outcome, the measure of precision and reproducibility that the assay has. These are the sorts of elements that you should consider in the design of a medical device. And so in summary, fully specified assays are needed so that data can be analyzed. These sorts of studies are required to understand if the data is interpretable across sites, if we've got a consistent, understandable definition of what it is we are looking at. Validated tools to assess outcomes are critical, and we recommend early interaction with the agency. And CDRH does provide assistance without validation study designs through the pre-submission process. The link here is to our guidance, and if you have any questions at all, please feel free to email. Thank you.
All right, I'd like to thank Jennifer for that overview. So you've heard from Jennifer on the regulation of assays for potential markers from the standpoint of adequacy of performance. You've heard from Vishal on a conceptual framework for validation of potential surrogates. We're now going to hear from Daniel Sargent, the Ralph and Beverly Calkins Professor of Cancer Research at the Mayo Clinic, on some of the major biostatistical considerations for demonstrating surrogacy, including, as we mentioned before, that just a correlation between the potential surrogate and the outcome of interest isn't enough. This is really more about validation of individual level predictions. So Dan, we're very pleased to have you with us today. We're going to talk about not only a general framework here, but some very important work that Dan himself has done in this area. Thank you.
All right, good morning, and it's really a pleasure to be here and see so many colleagues from NCI and FDA and the Alliance and industry, and very, very important topic today. So don't worry, there's not a lot of statistics in this talk, even though it is in the title biostatistical, but really talking about concepts here and things that we need to keep in mind as we then go through the rest of the day and think about using MRD as an endpoint in a clinical trial. So that's really, we're not talking about prognostic ability, we're talking about developing drugs and using this as a clinical trial endpoint. So we, I think, all know what surrogate endpoints are and true endpoints are, but just to make sure we're all on the same page, we do clinical studies because we're interested in true endpoints that really matter for patients, impact how they feel or how long they live. So overall survival, progression-free survival, patient-reported outcomes are really the endpoints of interest. But we know that these are difficult to use in clinical trials. They may require long follow-up, they may be rare, they may be costly to measure, and therefore we are looking for earlier endpoints. We would like the ability to use an earlier or a surrogate endpoint that could be easier to measure. Often we mostly care about quicker to measure. We want to get therapies established, their benefit, and get them to patients soon. So the goal of using a surrogate endpoint would be to either reduce the duration, the size, or the cost of a trial, and yet still provide the convincing inference, the convincing conclusions at the end of the day, so that when we are using a new therapy based on that endpoint, we have a strong understanding and a strong knowledge that these endpoints will reflect the benefit on the true endpoint, which is always what we care about. So really, this is the most important concept in terms of validation of a surrogate endpoint from a biostatistical perspective, and that is that the effect of the intervention on the clinical endpoint is reliably predicted by the effect of the intervention on the surrogate endpoint. So we're talking about an intervention, a treatment. And what is the treatment effect that we observe on the surrogate, and what is the treatment effect that we observed on the true, and can the treatment effect on the surrogate predict the treatment effect on the true? And that, if we can demonstrate that, and I'll talk about how we can demonstrate that, if we can do that, then we would feel very confident, at least within the setting that we explored, and I'll talk about the limitations of drawing inferences across different diseases and even different states of the same disease. If we can be confident that we can predict the treatment effect on the true endpoint from the observed treatment effect on the surrogate endpoint, that would be considered a valid surrogate. So there are two levels of surrogacy, at least in the last 15 years or so, the field has come to use these two terms of surrogacy. One would be surrogacy at the individual level. Surrogacy at the individual level is necessary but not sufficient. That would be that the surrogate predicts the clinical endpoint within an individual patient, that a patient that has a good outcome on the surrogate will have a good outcome on the true endpoint. There are lots of endpoints like this. This is a correlation. This is not necessarily a treatment effect. The more important consideration, the more important factor is the trial level surrogacy, which is that the treatment effect on the surrogate allows a reliable prediction of the treatment effect on the true endpoint. And statistical methods that have been developed estimate both of these quantities. There are a lot of endpoints that do well on the first measure, that they predict well at the individual patient level, but where it's much more difficult is to predict at the trial level. That is, the effect of the intervention reliably predicts a difference between treatment groups in, say, a randomized trial. So individual level association is not sufficient. There are many intermediate endpoints that are related to long-term endpoints like PFS or OS at the individual level. We've known this for 50 years. Patients who respond to treatment based on any response definition tend to live longer across many, many diseases. But this may well just reflect either the natural course of the disease or the effect of some specific treatment on the course of disease. It does not necessarily and directly follow then that a treatment that impacts that intermediate endpoint will similarly impact the long-term endpoint in a reliable and predictable manner. And that element of prediction is critical here. We need to know what level of benefit on the intermediate endpoint, on the surrogate endpoint, would predict a clinically meaningful benefit on the true endpoint. So it's even more than just correlation or even causation, so to speak. It really is also being able to reliably predict that treatment benefit, because we may see if a large benefit on the surrogate predicts some benefit on the true, but it's not a clinically meaningful benefit on the true endpoint, then that surrogate endpoint may still be validated in a statistical sense, but not be clinically useful if it doesn't predict a clinically meaningful benefit on the true endpoint. So this has been recognized for a long time. This is really one of the landmark papers on this topic by Fleming and Demetz from 20 years ago now, talking about what are the reasons why individual level association can fail to relate to trial level association. And I won't go through every one of these, but for example, in case B, we have a disease on the left, we have a surrogate endpoint in the middle, we have a true clinical outcome on the right, and the surrogate is on the pathway to the clinical outcome from the disease. but there may be multiple pathways from the disease to the clinical outcome, and it could be that the intervention only affects one of those pathways that is assessed by the surrogate endpoint, but the disease may have multiple pathways by which it impacts the true clinical outcome. So this could be a case where an intervention could cause a apparently meaningful change on the surrogate endpoint, but not translate into a change to the true clinical outcome because the disease has multiple pathways by which it affects the true clinical outcome. And there are cases here that can result in false positives for a surrogate. There are cases here that can result in false negatives for a surrogate. For example, case C is a case where the surrogate endpoint is related to the true clinical outcome, but there's an intervention that actually affects the true clinical outcome, but it doesn't do so in a way that's mediated through the surrogate. And so each of these conditions we have to consider every time we think about a new endpoint as a surrogate, not only the statistical perspective, but this is about the disease. This is about the biology and how fully does the proposed surrogate capture the intervention or the effect of an intervention on a true clinical endpoint. Does it capture the full causal pathway, or may it only capture part of the causal pathway? So the field has moved to what is called meta-analytic surrogacy evaluation. The initial work in this area, starting in about 1989 with the work of Prentice and through much of the 1990s, was based on single trial evaluation methods that have now been demonstrated to have challenges both from a statistical perspective. I won't go through all of these, but you can note in the literature the challenges in terms of unrealistically high treatment effects that are required or large sample sizes. I think more importantly, from a single trial perspective, limitation is the medical and practical limitations of this, that any single study may have idiosyncratic or chance associations between a true and a surrogate endpoint, and so we really need to demonstrate this across a number of studies in any given condition. So that has led to the use of meta-analytic methods, which allow, most importantly, the ability to have a direct inference at the trial level. That is, we can look within a randomized trial, what's the difference between two treatments on a surrogate, what's the difference between two treatments on a true endpoint, what's the association between those, does a benefit on the surrogate predict a benefit on the true, does the lack of benefit on the surrogate predict a lack of benefit on the true? Both cases are important. We need to, it's statistically would even be important if we can to show that a negative impact on the surrogate would predict a negative impact on the true. We hope we don't have very many trials like that. We don't want to be doing trials that generate negative outcomes, but of course they do, and if they're available, we should use those in our evaluation. The other advantage of the meta-analytic approach is that it allows inferences within a class of treatments. It may be that not every trial tested the same drugs, right? But they may all be from a similar class of treatments. They may not be the identical patient cohorts. They may have differences, some modest differences in eligibility criteria or disease stages. And if we really can show across some heterogeneity in the types of patients and treatments that are used, and we can demonstrate robust surrogacy across that entire spectrum, that helps us have confidence that in the future that same relationship is likely to hold, and it's not just for this one specific treatment, this one specific type of patients. I'll come back with cautions about that later on. So what are the key principles of the surrogacy evaluation through the meta-analytic approach? One of them is we need to have individual patient data. We need to get the raw data from each of these individual trials. Why do we need that? Well, we need to be able to consistently define our endpoints. We need to be able to consistently define our time scales. A lot of trials, when is time zero? Is it diagnosis? Is it date of randomization? Is it date of some surgery? Is it the date of initiation of induction therapy? Is it the date of initiation of maintenance therapy? All of these things differ between different trials. And when we get the individual patient data, we can set a consistent time zero, we can set a consistent hopefully also definition of endpoints, because there are a lot of different ways to define event-free survival. What's an event in event-free survival? And if we have the raw data, we can consistently define that across all of the different trials. The individual patient data also allows us to do subgroup analysis. What about the surrogacy within this subset of patients? Maybe the elderly, maybe the less invasive disease, maybe the earlier or later disease. And then also sensitivity analysis. What happens if we take out one trial or two trials, or we take out a group of patients that we may think is a little bit different, or maybe we think there's a specific group of patients driving an association. Well, we don't want an association just by a small group of patients. What if we take those out? Is the association still there? Having the individual patient data allows us to do this. And then, of course, it allows us to examine unpublished endpoints. We may be interested in endpoints that were not part of the original publication, and having the individual patient data allows us to do that. So really, that individual patient data allows so much more of a robust analysis. And fortunately, the field of clinical trials has moved so far in the last few years with sharing clinical data properly de-identified with patients who've provided consent, of course, but the ability for groups to gather this data at the individual patient level, and I'll come back to talk about this in a little bit, really has taken a major step forward over the last several years. The meta-analytic surrogacy evaluation, second key principle is the consistency of endpoint evaluation across studies. And I know that's an issue in the MRD field. What is the definition of MRD? What assay was used? What are the criteria? We need to understand that. We hope that's not my area at all. I'm sure we'll talk about that a lot today. But how consistently have these endpoints been defined? We don't want to pool together a bunch of trials that have very different definitions of these endpoints. Then we will have a very hard time understanding what the results mean. Inclusivity is another important principle. We do want to include all possible trials. We want trials with positive results, with null results, with negative results if they're available. I would say, though, that the failure to obtain all possible trials, I do not believe is a fatal flaw, as long as the trials are not selected based on the results of the trial. So when we do this work, and my group has done several of these analyses, we try to be inclusive, but sometimes, for whatever reason, a particular trial is not available, or maybe the data quality within a trial is inadequate to allow it to be used in the validation. I don't think those are fatal issues, because we're truly looking for consistency of treatment effects across the surrogate and the true across a span of trials, and not necessarily losing one or two trials is going to invalidate that inference. And then finally, if we want to do this from a formal regulatory perspective, it adds a whole nother level of intensity to this process, where we're going to want to pre-specify what are the conditions for trials that we will include in our analysis. What are the endpoints that we're going to care about, that we're going to look at? What is the statistical and analytical approach that we're going to use? It's not appropriate to just gather hordes of data and then go on a fishing expedition to say, oh, we found this endpoint correlates. Well, did you look at 17 endpoints? Well, that's not very convincing. Much better is say, we believe, based on some individual studies and preliminary work, that we have this endpoint we think is promising. We have this as the true endpoint. We're going to use this statistical approach. We treat it as though really it's a prospective trial, but it's a prospective meta-analysis done to establish fully a surrogate endpoint. So this approach has been used in many different diseases. These are all solid tumors. I'll give an example later of a lymphoma example, but we see colorectal, prostate, and breast cancer have been examined through this approach. We don't care about the details because solid tumors is not the name of the day here, but it has been used quite often in many different settings. So I want to go, this is kind of the only kind of statistical part of the talk, so forgive me, but I think it illustrates some important points. How do these measures work in the real world? We have to understand that. We can't be in an ivory tower and say, this is how you should do it. Yeah, it's really hard, but we need to know, does this really work? So we performed a major simulation study on mimicking oncology clinical trials with a primary endpoint of OS, a surrogate endpoint of PFS, but really the choice of the endpoints doesn't matter. It could be MRD, it could be anything else. Assumed we had multiple trials available and assumed that each trial had 2 treatment arms. So we made up a bunch of trials, and we wanted to see how well does the the meta-analytic surrogacy approach work in different settings of those trials. So what are those settings? Well, is the true trial level association, which we measure by something called R-squared. It is a value that goes from 0, which would indicate no association between endpoints, to 1, which would mean perfect association between endpoints. So we looked at it at values of 0.2, really bad surrogacy, 0.6, kind of modest surrogacy, 0.9, very strong surrogacy. We said, what happens if we only have 6 trials, which is about the limit of where we could use this method, 18 trials, which would be great, or 36 trials, which any of us would love. Very difficult to have that. How about the sample size within trials? Is it a relatively modest trial of 500 patients? I know maybe in some diseases that's not so modest, or a large trials, do we have censoring of our patients? And importantly, the range of treatment effects. Do we have tightly grouped treatment effects, or do we have a large group of treatment effects? And we looked at a number of different approaches that have been proposed for surrogacy validation, different statistical approaches. The reality is there really wasn't much difference at all in the statistical approaches. Very sad to see that. All of our statistical work didn't really make that much of a difference, but nonetheless, that's what we do. So what's shown here? The different colors don't matter. Those are the 4 different approaches. And what you can see is all the approaches pretty much said the same thing. So that's not the important part. This is 1,000 simulations of 6 trials or 18 trials or 36 trials. What's shown here is when everything's at the good condition. We have a lot of trials, we have high patient numbers, we have a big range of treatment effects. On the left, the true R-squared value was 0.9, you can see at the bottom. And the estimated trial level R-squareds were very close to 0.9 on average, you can see on the left. In the middle, the truth was 0.6, and our estimates, there was some variability around it, but in general, the estimates were right on the target at 0.6, so the statistical methods were working. When we had low surrogacy on the right, the truth was 0.2, and we were estimating 0.2. Still sees there's definitely variability there, but the methods were calibrated correctly in the perfect sense, in the perfect setting. What about when we vary the number of trials? The one on the left is 36 trials. We already showed that. That's the good setting. When we went to 18 trials, we still did pretty well. You can see the estimates were right on average correct, and there wasn't that much variability between the simulations. But on the right's where it starts to get a little dicey. We do see that's with 6 trials. Suppose we do a meta-analysis with 6 trials. We do see on average, that's the little black bar in the middle of the color, on average we're okay. But we have really long tails on this distribution. We have a lot of cases where even though it was truly strong surrogacy, 0.9, which is good by anybody's standards, I think, we see a lot of cases where the estimated surrogacy was much reduced. So this indicates the risk. Doesn't say don't do it, because on average we're not biased. On average we're okay. On average we estimated the surrogacy correctly. But there's a big risk when you have a small number of trials that you will underestimate the surrogacy using this approach. I think that's something we just all need to keep in mind and why it's so important to get as many trials as we can. This one's also very important. What we're looking at here is the range of treatment effects. On the left is a large range of treatment effects, meaning we have some trials that are positive, some trials that are intermediate, some trials that are negative for the true endpoint. When we have a large range of treatment effects, we actually have good estimation. That's on the left. But in the middle and on the right, that's when we have a small range of treatment effects. That is, we have treatment effects that are clustered either around one or even when there are a bunch of positive trials. On the right is a bunch of positive trials. We see there that the surrogacy estimation is actually biased. It's biased in the negative direction. The reason is that these measures really look, from a statistical perspective, there are R-squared measures. They're looking at consistency of effects across a range of effects. When you don't have a range of effects, it's really hard to estimate. You think about drawing a regression line, a line through a cluster of points. When the cluster of points is spread out, you can draw a line through that pretty well. When the cluster of points is tight, that line has a lot of variability in it. And that's what's happening when we get this small range of treatment effects. So that's why it's, again, critical to get the positive trials, the negative trials, and not just a convenient sample or trials that have been published in big journals that all have positive effects, because they could really bias your results. The last specific data I'll show is what happens, okay, so say we only have our 6 trials. Well, what some people do sometimes is they create artificial units. They say, let's take our 500 patients and let's split them up into littler groups. So it looks like we have more trials. We don't really have more trials. But instead of one trial of 500 patients, let's pretend we have 5 trials of 100 patients. Instead of one trial of 500 patients, let's suppose we have 5 trials of 100 patients. Shouldn't that solve our problem? Well, the results here said that did not help. The true R-squared is up at the top, 0.9, and even when we generated patients within centers, because that's what people say sometimes, well, we have a few big centers, let's treat that center as an individual trial. That was on the left. When we used the center that the fake patient was drawn from and estimated the surrogacy, we were biased low. When we did random splits, that is just randomly split, we did even worse. You can see those numbers are even lower. When we actually just did the analysis with the 6 trials we had, that's on the right, you can see that the bias was much reduced. On average, the bias, there was a little negative bias. These should be up at 0.9. They were a little bit biased. The variability still was high. We've actually seen the one on the right. That's just from a previous slide. But what it says, unfortunately, I wish the results were different, but split, just taking a small number of trials and splitting them to make more mini trials is not the way to go. Didn't help us, at least in this situation. So the conclusions, very briefly, when we have a large number of trials, when we have a large effective sample size, when we have a range of treatment effects that's high, we can do really well with this approach. When we have not those optimal circumstances, the surrogacy estimation has high variability and fortunately is biased in a conservative direction. It always showed that the surrogacy on average was estimated lower than it was true. So what that means is, yes, if we do it, we don't really, I believe, have to be worried about false positives so much. We do have to be worried about false negatives. That is, we run a substantial risk that the surrogacy would be underestimated, but on average, that's much better than if we had on average a overestimation of surrogacy. That we really would be nervous about. And for the small number of trials, estimating surrogacy based on smaller units did not really help. We have another follow-up paper that's referenced there about what are specific cases where we might be okay to do some amount of splitting, and random splitting is not a good idea. Okay, so I want to go through a couple case studies that we've done. First one I'll go through really quick because it's colon cancer, and you guys you're not colon cancer people, but we did do an analysis like this in colon cancer, looking at a very boring endpoint called disease-free survival. Not sexy at all, but it's an important endpoint. We know that patients who have stage 2 and stage 3 colon cancer, most of the time before they die, they recur from their disease. So we had a hypothesis that we could look at early endpoint of disease-free survival to replace overall survival as an endpoint in colon cancer trials, stage 2 and stage 3. We gathered data from a large number of randomized trials. This is the list of the trials. You can see everyone was kind of salivating. We had 43 treatment arms and 21,000 patients. Things did really, really well. And this is the single slide you should always look for in a meta-analytic validation. What's the treatment effect on the surrogate? On the x-axis, what's the treatment effect? On the true, on the y-axis, here we've got a good association across the range of treatment effects, positive treatment effects, null treatment effects, and in fact negative treatment effects. Second case study is the FLASH project in follicular lymphoma. So follicular lymphoma is a disease where the now clinical course is very extended. Patients have long progression-free survival, fortunately medians of 7 to 8 years, and we set out on a project to more reliably assess or to try to demonstrate an earlier endpoint. We created a academically led collaborative group called FLASH to perform a prospective meta-analytic evaluation. And this one we did more formally. We met with regulatory authorities multiple times throughout the course of this project to get, hopefully, buy-in on our approaches. Our hypothesis that patients would have, that had a durable, complete response would have better endpoints on progression-free survival, and we set a specific hypothesis that the treatment effect on the CR rate at 30 months following initiation of therapy would predict a treatment effect on progression-free survival. We used the measures that I've discussed here. We pre-specified in that third bullet what the criteria would be for us to consider appropriate surrogacy, which is that the R-squared measures had to be at least 0.8 with the lower bound of their confidence interval greater than 0.6. We set out pre-specified inclusion and exclusion criteria for what studies could be included, and we were able to demonstrate a very strong association between these two endpoints, both on two different measures. That's a technicality, R-squared weighted least squares and R-squared copula of 0.88, 0.86 with confidence intervals that did exclude 0.6. And we concluded, and we are continuing our discussions with various agencies about the success of this approach, but it was an approach with 13 trials that we were able to demonstrate a valid surrogate in follicular lymphoma. So what are some limitations that we must keep in mind? And I talked about some of these already. The endpoint definition, the endpoint must be consistently defined across the included studies, and that's something I know we'll talk about today. We need to worry about classes of agents and agents that work by mechanisms that are different than those that were included in our meta-analysis. We cannot necessarily conclude that the surrogacy will hold. Clearly, surrogacy is disease-specific, but even extent of disease-specific, and it may work in patients with late-stage disease, but not in patients with early-stage disease. And it is very sensitive to the time era and the state, the nature of the patients that are being treated. And what does the survival after the surrogate look like? How many subsequent effective therapies might be available? To show how it's maybe sensitive to the stage of cancer, here's an example in colon cancer. On the top is patients with stage 2 disease. On the bottom, patients with stage 3 disease. And we show that even in relatively similar patients, we had strong surrogacy in stage 3. On the bottom, we had poor surrogacy in stage 2 on top. We need to think about the time era. So here's colon cancer progression-free survival in the metastatic case in an old set of trials, trials before 2000. Strong surrogacy between progression-free survival and overall survival, R-squared 0.98. That was published in 2007. We redid this analysis using newer trials published in 2014, and our R-squared fell to 0.54, mostly, we think, because now there are effective therapies available for patients with second and third and later lines in colon cancer, and that impacts the relationship between the surrogate and the true. I'm going to, in the interest of time, I'll skip this because this is a nice, Don Berry, Don, there he is. I put this in just for Don, but I don't have time, sorry. So what can we do when we don't have so many trials? Well, we need to fall back on our understandings of biology. If we can eliminate back to that causal pathway diagram of Fleming and Demetz, if we can better understand disease pathways, we can eliminate some of those risks, some of those uncertainties. We can do individual patient modeling. If we can do robust individual level association across many treatments and over time, it does reduce our risk and concern about confounding. And I think we may well have to do iterative analysis, and this is what we've done in colon cancer and what we're continuing to do in lymphoma. Look at the trials we have, assess the association, and then update those as new trials become available, recognizing that if we wait for the perfect, we may be waiting too long. We may need to make progress right away. We've been able to accomplish this through academically based collaborative groups where individuals agree to contribute the data. We have a coordinating center for data analysis and management and statistical analysis where we use the data only for what the parties agree to provide the data for, and we as academics are generous with our authorship. So this is the last slide, key principles for surrogate endpoint validation. I think the meta-analytic multi-trial approach is really the state of the art today, based on individual patient data, a prospective analysis plan, supported by strong clinical and biological evidence. And we have to understand this challenge that I just briefly touched on, that if a surrogate is established and then it loses its surrogacy, what does that really mean? Does that mean that endpoint is no longer reliable? Or does that mean we still have demonstrated our proof of principle and the reliability is now less because of things that happen after that endpoint, but we could still comfortably conclude that changing that surrogate is still important on that disease pathway and is important? That's a question for the field, and it'll differ, I'm sure, by disease site. So with that, I thank you, and I think we have some time for questions and discussion.
Thanks very much, Dan. And we do have some time for questions. If you all do have a question, just raise your hand. There are microphones on the table. Maybe while you're thinking about those, Dan, you talked at the end about the importance of understanding of the underlying biology as potentially helping this process. Is there a formal way to incorporate that in the analysis that you do, or is this more just helping to guide where the effort should be focused?
Well, that's a great question. There are statistical approaches we could use, multi-state modeling approaches. As statisticians, we always get a little nervous about them because they do rely on assumptions, and so really our faith in them is based on your faith in them, and do you really believe these disease models? If we can be confident we understand the biology, there are approaches we can use to integrate that data more formally. I would say that is work in development. There's not a formal approach to do that at this time, but it's something we need to pursue, absolutely.
No questions? Okay, I can keep going, but please go ahead. And microphone, please. Well, it's for the recording, so need your voice for posterity. Thank you. And just push to talk, and you can lift it up.
Hi, thank you very much for a very nice presentation and for all panelists. My question maybe first directed to Dr. Beth Nanger in terms of the agency's intent to explore using MRD as a surrogate endpoint to support accelerated approval, and in the case of it's reasonably likely to predict clinical benefit. And once I heard Dr. Sargent's presentation on validation of surrogacy, I just want to get clarity from the agency. In order to use this in accelerated approval, is this the level of validation you're looking for? Or with such meta-analysis exercise, you will actually consider MRD as an endpoint, as an established endpoint to support regular approval? So maybe to FDA first.
To not paint with such a large, broad brush, I can't say specifically. I think it's going to really come down to the disease-specific areas. And as I mentioned, we need to ensure that MRD is a reliable surrogate endpoint that's on the causal pathway. So I would encourage you, if you have specific questions about a specific disease, the disease-related panels may specifically answer the question about MRD in the context of each disease.
I mean, that is a major point of the panel discussions today, is to try to bring to each of the diseases the kind of frameworks that you've heard about this morning. Just one related follow-up, though, maybe for Dan. You went through in your examples some specific criteria for what constituted surrogacy, for example, R-squared in the 0.8 range and so forth. Does that seem like a generally accepted range? What is the level of consensus around what the criteria for acceptable surrogacy should be?
Sure, that was one of the most challenging, or let's say, interesting discussions we had with the agencies, is what is the proper threshold, and there's no fixed level. We base it on established literature and kind of conventions across a number of different diseases, and we knew that in settings where we had high levels exceeding 0.8 most of the time, that was accepted as a valid surrogate for even full regulatory approval, such as for disease-free survival in colon cancer. When there were settings where it was observed in lower range, 0.6, even 0.7, often the literature would say this is a promising but not validated surrogate. 0.8 we picked, it seemed reasonable. The agencies, both EMA and FDA, seemed to be comfortable with that. Of course, you know, I'm just saying that. They could speak for themselves. It also really does depend, though, on that confidence interval, right? Because if you have 0.85, but your confidence interval goes down to 0.6, that's not very convincing. It also depends on the prediction effect, and that how reliably we can make that prediction. Because maybe if you have an association that's lower on that R-squared scale, maybe it's 0.6 or 0.7, but it's still reliable enough that you have a big treatment effect on the surrogate, it's still going to predict a treatment effect on the true, maybe with some more variability. But if you have that good prediction, even with variability, that still may be an acceptable endpoint. Depends on the disease and the amount of variability. But that's another topic I didn't talk about, which is a concept. It's called surrogate threshold effect. That is, what effect on the surrogate do you need to see to reliably predict a treatment effect on the true? And so even if your surrogate's not great, but it has a meaningful surrogate threshold effect, where if you see a big impact on the surrogate, you can be confident. If you see a modest impact on the surrogate, maybe you can't. That's something we need to think about. So it's never just one number to think about. I focused mostly on the R-squared, but also the surrogate threshold effect is very relevant.
Russ, please go ahead. Microphone two.
There you go.
Works now. So, question to Dan. So early in your talk, you mentioned two potential problems or issues with surrogate endpoints or validation of them. That one is that it depends on mechanism of action of the particular type of treatment. And second, then, as new treatments, subsequent lines of therapy are introduced, relationship with OS especially can be affected. Mm-hmm. And then of the talk, you mentioned that so once you establish a surrogacy, and then it potentially goes away, presumably because of subsequent lines of therapy. Do we really, give up on that, or as it presume that it's, as a matter of principle, it's established? So, but there is still another possibility that we move from different types of treatments. So it might go away, not only because of subsequent lines of therapy, but for this type of therapy, it might not be a surrogate. So can you comment on how potentially try to distinguish between the two reasons for surrogacy and not being surrogate anymore?
Well, I might ask for your opinion on that as well. I mean, that's a certainly very challenging situation. It's not something we can statistically identify. That really requires our understanding of the pathways and does this treatment impact the surrogate and the true in a similar mechanism that the previous treatments did, or is it going after a totally different pathway, totally different mechanism, so that with this type of treatment, the surrogate is not going to be reliable, right? And so it's probably more of a medical and biologic consideration than it is a statistical consideration, is this new treatment expected to affect the true endpoint through the pathway that's measured by the surrogate or not, and that's really the key consideration there. If we think it is going to affect the surrogate and the true in the same way, and we can feel confident in that, then maybe if the surrogacy goes away in trials that have later, therapies available, we'll feel more confident in it. But I think it's likely not a statistical issue. You may disagree with me. I think it's more our understanding of the pathway and of the new treatments.
In the back.
First, many thanks to the three presenters for your clear and concise distillations of the fundamentals on this topic. Question for Dr. Sargent. Could you reflect on your level of optimism about the degree to which this approach you've described of a meta-analytic analysis of multiple trials can be extended to hematologic malignancies in which the incidence typically is much lower than that of surgically cured colon cancer? The disease presentations are heterogeneous even within that reduced incidence, and the measurements of minimal residual disease are either non-standardized or their implementation is non-standardized. Thanks again.
Sure. I mean, that's a great question. So the disease, so conducting a meta-analysis based on trials that are 2,000 patients or 300 patients is not really a critical distinction. We can do the meta-analytic approach using randomized trials that can be much smaller. And when we did this in the FLASH group, the trials were much, much smaller. We had 13 trials, and together we had about 5,000 patients. So most trials were in the range of 300 to 400 to 500 patients. There was, I think, one that was 1,000 patients, and we did successfully establish a surrogacy in that case. So I'm not concerned so much. Of course, more numbers, bigger is better, but we can do this with smaller randomized trials. In terms of heterogeneous disease populations, that is a concern, and we have to understand that surrogacy, as I discussed in colon cancer, but also within the FLASH, we spend a lot of time in FLASH looking at Ann Arbor stage and Flippy score and demonstrating that we had surrogacy across different prognostic groups within that disease. So I think that is something we can do when we get the individual patient data. It's something that we have to do, I think, to convince ourselves that the surrogacy is robust. Your last point is the most difficult one, I think, and that is when the surrogate endpoint has not been reliably, or no, I shouldn't say reliably, it's not been consistently measured across the various diseases. And then I can't answer that very well as a statistician. I don't know if these are meaningful differences in the analytical approaches or the measurement approaches. If they're all measuring the same thing just a little bit different ways, that's something I truly don't understand, and I hope to learn a lot about that today. To the extent that we're variations on a consistent principle, I feel relatively confident. If we're truly measuring different things, then I'm not very confident. The question is, do we have the data? Are samples banked? Are they available that we could go back and try a consistent approach that maybe wasn't used in the original trial, but if the sample's there, we could try to agree on a standard and assess that across multiple trials? That might be a promising approach. I don't know the availability of that. So I think that's the most important variable is in order for us to really be confident in our conclusions is, are we measuring the same thing, and is our surrogate defined consistently across these trials?
And Dan, just to follow up on these points, based on your experience, I know you talk a lot about solid tumors and not hematologic malignancies. What do you see as the best opportunities for increasing the feasibility of developing better evidence on surrogacy. You've talked about clinical networks coming together, use of common data models. Any particular points you want to emphasize that could be relevant here?
No, I think you're capturing it. I think the FLASH really was a very successful, and we're actually doing the same thing, or a very similar thing in diffuse large B-cell lymphoma right now. We've got a couple abstracts submitted to ASH on that topic, looking at different endpoints. The critical aspect there has been cooperation between various pharma partners, between various academic groups, European, U.S., and really coming together for the good of the field. And I think that's what it takes. And the field of data sharing and acceptance of data sharing, and the moonshot and big principle is data sharing. I hope we're at a time that this community could come together and agree to do that and really provide that raw individual patient level data, which allows us to do these analyses maybe a little bit differently than they were done in any individual trial.
Yes.
We focused on...
You got to press the button.
We're focusing to this point on this progression-free survival, or things that we can publish papers, the papers can be published in journals. But in terms of thinking of surrogate endpoints, there's been no discussion of if the intervention you're doing changes the natural history of the disease. So you induce a mutation in a subset of patients where they die quickly.
Yep.
Or more importantly, say survivorship issues where you introduce a new therapy and at 3 or 5 years patients are getting osteoporosis or some other complication that greatly impairs their life. So I mean, this is more of a regulatory question. So as we're coming up with thinking of surrogates that we're going to use for trials, do we need to incorporate, because how trials go, they go from phase 1, 2, 3. And often the data about these questions, how resistance develops, how the natural histories change, what are the late-term complications, come from phase 1 and 2 trials of new drugs. And so how do we need to consider incorporating the spectrum of data across the development of a drug in terms of coming up with individual surrogate markers for therapies? Because that's what we're here to do.
I think that's an interesting question and challenge that we face. Looking at phase 1 and 2 trials, unfortunately, we're limited by sometimes population sizes. And then to top it off, there's heterogeneous populations, so using a meta-analysis approach sometimes is inappropriate. That being said, in specific disease areas, we have seen that approach used, but the more data we get, unfortunately, sometimes a year later, that may be an outdated approach. So we're our hands are just as tied as sometimes yours are. So, but we're always open to looking at that information. We're always open to assessing the benefits of approaches.
Dan, maybe a follow-on, but turning that around a little bit. What about incorporation of data from single-arm trials or non-randomized settings? How can that contribute to the surrogacy determinations?
So I'll give you two answers. My formal statistical answer is that data doesn't help us very much. We can't use that in the meta-analysis. The meta-analysis is really based on estimating a treatment effect. And if we have a single-arm trial, we can't really estimate a true treatment effect. That is a difference between an experimental treatment and a control treatment. Now, having said that, my group and others are examining alternative approaches that we know we need to take advantage of that data if we can. It often is used, crudely at the individual level, association and understanding within patients, the pathways. If we are comfortable with that, that helps our maybe meta-analysis be more reliable, more believable if it's only based on 6 trials instead of 12. But at this point, we don't incorporate that data into the formal meta-analytic evaluation.
Okay. Dan, I'd like to thank you and our other speakers this morning for a very helpful opening and introduction to the topics that we're going to discuss today. Thank you very much. All right, we are now going to take about a 15-minute break. Slightly less than that, we'll be reconvening at 10:45 for our next session.